So with no further ado, please help me welcome our distinguished guest, Dr. Shyamanga Borooah.
Thank you so much, Vicky. That's very kind for the long and detailed introduction. So hopefully, this talk was scheduled sort of about 3 or 4 months ago, and I was talking about a number of my research areas. I'm a retinal specialist at UCSD and run a retinal degenerative clinic. And beyond a shadow of a doubt, Vicky said that the biggest amount of interest was in stem cells, and quite applicably. So this talk today is really to update you on research in stem cells, and also to talk about a couple of cases, and just to have hopefully a bit of an understanding of where we are and then where we hope to be in terms of stem cells. So I'll just start the presentation just now. So hopefully you can see this.
Looks good.
Yeah, good. So just going to move. There we go. Okay. So what will I cover today? So I'll briefly go through sight loss. I'll explain what a stem cell is. Then I'll review the latest research with small clinical cases along the way. And the areas we're really looking at here are stem cells modeling, which is a small area, but the main areas are really replacement and regeneration. So without further ado, let's carry on. And really there's a real need for this. The cases of blindness in the US have gradually increased. So from 2000 to 2010, they've increased by almost a third. And the predictions are, these are figures from the National Eye Institute. You can see that 2030, 2050, we expect this to increase further to well over 7 million people. So, there's a real need for treatments for blindness. So what are the main causes of blindness? And research from this really has been relatively lacking in the US, but the main causes are very similar to where they were. This paper is in 2004, but we have cataract, which is a treatable condition. And there's obviously refractive errors, which can be treated with glasses. But the most common cause of sight loss is macular degeneration, and in addition to that, glaucoma. And there's a difference amongst different populations, but there's an increasing amount of diabetic retinopathy. The great news is, with a number of these conditions, such as wet macular degeneration and diabetic retinopathy, there are increasing amounts of new treatments, and these are injections into the eye which stop blood vessels forming. However, there's a whole range of conditions, and especially the dry macular degeneration and glaucoma, where there aren't conditions after the sight loss has occurred. So why is it so important? Well, I'm really speaking to the converted already, but really it's such a debilitating condition because vision is the main sensory system for humans. So there's a lot of independence, and multiple studies have shown that there's effects on physical and mental health. But why is it important for other people? Well, there's a massive burden on social services and on care. It affects people in terms of the employment. So employment levels are reduced. So people with sight loss are obviously less likely to have employment. And overall, this increases costs in terms of healthcare and tax. So actually, everyone should be looking at sight loss. And this has really come at the right time in terms of funding, because with the recent elections, the Proposition 14 was accepted. And this looks at the California Stem Cell Agency, which tries to bridge basic science and tries to get into the translational side to try and give treatments. And vision is really one of the core diseases they're trying to look at. So let me briefly go through how we see, and again, this may be pretty basic for some of you, but I'll just quickly go through this. So essentially, the eye is almost like a camera, or one of the old cameras, where you have a lens, at the front of the eye, which focuses light at the back of the eye. And the central part of the vision is called the macula. The macula is the main area for color vision, and also the area that you use to read, and also look at people's faces, for instance. So I'm going to try and run a short video here. I'll try and explain this for people who can't see, but basically I'm showing that the light goes to the back of the eye, and then we're looking really at the macula. And the central part of the macula is called the fovea. So the macula is probably the most important thing you have to remember. And if you go into the macula and the retina, there's a set of cells which converts the light energy into electrical signals, and they're called photoreceptors. Underneath that layer is a layer of cells called the retinal pigment epithelium, RPE, and they're a black layer of cells. And they're situated on something called Brooks membrane, which is a foundation. Underneath that, there are blood vessels. And for sight to occur, you need all those layers. So I'm just showing a picture now of a retina cross-section. And basically, light, it took me a long time to understand this actually, but light actually goes through the retina. The retina is clear. So it travels all the way through the retina and then to the photoreceptors. And then it reverses. So it goes, the signals from the photoreceptors move back up through the retina and eventually move towards the optic nerve and to the brain where the processing occurs. And actually your main visual center is right at the back of the head. We talked about vision and how you have vision. So sight loss can occur in any of those areas. It can occur in something that stops the light coming in, or more commonly in the retina and loss of some of those cells. So most commonly degenerations go through stages. So that's the most common cause of sight loss. So glaucoma is a neurodegeneration. Macular degeneration obviously is a loss of RPE cells and then photoreceptors. So it goes through stages which are pre-symptomatic, symptomatic, and late-stage degeneration. And really, you need different kinds of treatments for different stages. So the things that I'm going to talk about today are really cell transplant for disease which is already progressing, and also cell supportive treatment, as well as cell transplant for late-stage disease. But one of the things that I want to understand, or I want to understand, is the development schedule, because one of the things that I get asked about a lot is, why are there not treatments already? These things have been developed in the lab, and it's still taking years to get to approval. And the reason is because, as you've noticed with the COVID side of things, there's multiple phases of studies. So with COVID, that was accelerated multiply, but usually this takes almost 6 to 5 years to get from preclinical to clinical trials, and then clinical trials can take anywhere between 5 to 7 years before a final process of about 2 years for 1.5 to 2 years for FDA approval. So this is a long stage between preclinical, phase 1, 2, 3, and FDA approval. So this is why stem cells haven't jumped straight from the lab to trials or treatments. But I'm going to sort of inform you about stem cells and how they've actually been involved in trials, and how we're getting close now to potential treatments. So what are stem cells? The most common questions are, how are they made and how do they work? So you may not know this, but this is someone called Ian Wilmot, who I was fortunate to work with in Edinburgh. And the sheep that he's sort of cuddling here is Dolly the Sheep. So everyone seems to know Dolly the Sheep, but there's a very little understanding sometimes of why Dolly was so famous. And Dolly was famous because basically they were able to take a cell from an adult sheep and then turn it back into a stem cell by placing it under certain conditions. So that's essentially what a stem cell is. So a stem cell goes back to almost a birth-like condition. So I'm just going to try and play a video here. So this is courtesy of Eurostemcell, some of these images, so I've just edited these. So stem cell has two essential characteristics: self-renewal, and the second thing is that it can differentiate into different tissues. So a basic cell is a stem cell. So it's like a building block cell that can form other cells. So differentiation means that they can go to different kinds of end tissues, such as skin, blood, and eyes. So we have these kind of cells all over the body. Even in the eye, in the front of the eye, we have stem cells already. And the skin, when you get cut, that has stem cells. And these suddenly grow. They're not basic stem cells, but they're a certain kind of stem cell. So the next big controversial topic is an embryonic stem cell. So what's the difference between this and the new type of stem cell called the induced pluripotent stem cell? So an embryonic stem cell is, as you suggest, it's an embryo being fertilized or an egg being fertilized. And those first cells are the embryonic stem cells. And these were initially found in the 90s. And when they were taken and grown, it was possible to grow different cell tissues. So now, these cells are still used, but now we're moving on to a different kind of stem cell, which is the kind of stem cell that I've potentially or mainly been using. And these have a different kind of potential. So before, we could only go towards a definite end cell, but now we can actually reverse the process by adding a couple of factors. So it's almost, I don't know if anyone watches the Bake Off, but it's very similar to the Bake Off. With the right kind of recipes, you can actually turn an end cell to a stem cell. And these are called induced pluripotent stem cells. So pluripotent means you can make any tissue. Induced means it's been brought induced, that means made by these factors. So I just quickly show some work from my lab. I don't want to show all the data because we haven't published completely yet. We're about to publish some work. But essentially, development of the eye from birth takes about 60 days to form the normal shape of the eye. However, the retina is still forming. And stem cells essentially follow that line. So I'm just going to show you how to make stem cells. So this won't be a perfect recipe, but I'll show you how to do that. So essentially, this is what we did. We took skin samples, or actually more recently blood. This is the skin sample, and these are cells growing out from there. And on top of that, we placed these factors, which turned it back into stem cells. And these are actual stem cells, so little stem cell colonies. So these cells, if they're directed the right kind of way, can make heart, bone, or other cells. And we obviously made towards the eye. And the eye develops really essentially from the brain. And here, what we've got are sort of early neurons coming out of the stem cell area. And that's day 7. After day 28, so very quick, you've got these mini retinas, and these are covered in black cells, which are the cells that I'm interested in, especially, as well as the photoreceptors. And if you take these black cells and then plate them down, they make this lovely paving stone pattern. And these are the cells which are the ones which are affected initially in macular degeneration. So what can we use these cells for? Well, I'm going to go through a couple of examples now. So, these are almost an unlimited supply of cells. So you can use them for modeling disease and to try and get treatments. So you can test lots of treatments on these cells because you've got unlimited supply, unlike the human body. And then the other thing I'm going to cover, obviously, is transplants. So here's an example of modeling. So essentially what we did was we put those cells that I showed into a dish with a little membrane. And this essentially replicates the outer part of the retina. Cells which are diseased and control cells from brothers and sisters who are unaffected. And on the top, I'm showing a picture of a diseased retina staining for a factor, which is in brown here. On the bottom, I'm showing the cells, which are in blue, and the same factor is being stained, which is in green. So essentially, we're creating what's going on in the back of the eye in a dish. And if you look at this, the green is the disease. So on the left-hand side is are cells from a person carrying or having a disease, in this case, a form of macular degeneration. And on the right is control cells from a person who is normal, similar sort of age. And so essentially we're showing a marked change. So you can imagine if we can turn what's on the left-hand side to the right by testing different treatments, this will potentially be moved on to humans and clinical trials. And that's essentially what we're doing, and I'm doing in my lab, looking at this with treatments and also with gene editing. But we've really moved on. So as I said, we've not only just made these cells, but we can make almost whole organs. And it's now been almost 10 years since Yoshiki Sasai, who's pictured here, started making cells. So again, what I'm showing here, just for those who can't see clearly, is there's an early retina in green, but it has to fold. So what it does, it self-forms with different signals. And essentially what they're making are photoreceptors and the retina in a dish. So these hold great promise for modeling eye disease. Okay, so let's move on to other translational things, and really the area of interest for the group here, I think. So this is a very common case for my retina clinic. So a patient is 70 years old, healthy, but has noticed that central vision has gradually deteriorated, and real problems, especially with contrast and low light. The patient has given up driving and is finding real difficulty with reading now, and asks, are there any kinds of treatment or where research is? This is a macular degeneration. This is an example here from, and you can see this is the macula. This is normal retina, which is the pink color, but slightly paler here is the atrophy or loss of cells. And what does that mean? Basically, if you see this functional image, you can see even closer that the cells have been lost with time very quickly. And I'm showing a cross-sectional image now of sort of post-mortem retina. This is normal on the left, very nice structure, all the way to the outside, and the pigmented cells, the RPE cells I mentioned. And on the right are the damaged cells where they've lost RPE, and the structure is in a macular degeneration. So what essentially happens, so I made it very simple here. This top area is the retina, and the photoreceptors are in black. Underneath are the RPE cells, which support the retina. And in macular degeneration, in early stages at least, these cells are gradually lost. Eventually, the photoreceptors will be lost as well. So there's a very narrow window where they can potentially be saved. And so there are a number of trials to try and replace those cells. So I was fortunate to be part of one of these trials, and that's part of the Astellas and also the ACT type studies. And I'll just briefly talk about the other studies. So the kind of treatments I'm talking about now, or trials I'm talking about now, so these aren't treatments yet. Macular degeneration, can treat macular degeneration, can treat Stargardt disease, best vitelliform disease, and potential loss after things like central serous retinopathy. So these are relatively common diseases. So the first was in phase 1-2, so that's again early phase clinical trial, and this used single cells, and these were injected under the retina. This technique was tested in macular degeneration, dry macular degeneration, and in Stargardt disease. So what I'm showing here is actual footage from one of the operations, and these are very delicate. You can see already the amount of damage in the retina, but basically what's happened is a area of fluid is made, and in that area of fluid, we'll call a bleb, very tiny needle here is used to inject some cells. And so what happens to these cells? Well, from the study that I was participating in, you can see that at baseline, there's lots of areas of bare retina. And the injection took place in an area designated by a green square here, a square. And essentially a large area of fluid was made, that bleb that you saw. And these cells are the black cells, are gradually growing. So you can see here that the cells are growing with time, and also that suggests survival. And imaging and scanning also suggests that they're surviving quite well. However, the only trouble with this was, so the good news with this was that it appeared to be relatively safe, except in very high doses. And but the trouble was that single cells, although they survive, they don't form the right structure of the retina. So something else was needed, because this didn't improve vision, and it also didn't improve quality of life from what the patient said. So the next phase study, so that was from 2010 to 2014, but published recently. And then the next phase was to try something else, because the retina occurs in layers. The thought was to maybe try a patch, because then the RPE cells would be put in the right place in the gaps. So a phase 1 study was done by the London Project to Cure Blindness, sponsored by Pfizer at the time. They had 2 patients with wet AMD and delivered a patch. So I'm just going to, all the papers that I talk about are referenced below if anyone's interested to look at them. So the RP cells are very similar to the ones that I showed you that we've grown in our lab. And then they were grown on a special membrane. This membrane is transferred to another artificial plastic. And as you can see here, this is transplanted underneath the retina. So you can see the retinal blood vessels here. So this is the patient at week 4. So you can see some early pigmentation. At week 24, we can see pigmentation across the whole area and moving out. And by week 52, the area had grown even more slightly. So the majority of the growth had occurred in the first 2 or 3 months. So this is a very small subset of patients, but they found that delivery was successful, and there's actually improvement in letters. So it's quite exciting news at the time. And this improved by a couple of lines in both patients. And all that was used was local immunosuppression, that means steroid injections to the side of the eye. And this showed safety, lack of tumor formation of these artificial patches, and it was really early evidence. The only trouble with the positive results was that this is in patients with wet AMD who potentially would have been improved with other forms of treatment as well. So it's a bit debatable to see whether this is purely due to the patch or because of the AMD or the fluid has improved. But this is a good baseline study. So there's a further update to this, and this is published by Amir Kashani, who is just up the road at USC. And he's working with a group from UC Santa Barbara. And they've developed an RP patch, which is very similar. And basically in 2019, they started a phase 1/2A study. So again, these are early phases, which is safety trials with a bit of efficacy. And phase 3 is the real efficacy trial. So they started off and showed the results from the first 5 patients. And in these patients, they had dry AMD. The patch is very interesting. It's, you can see on the right-hand side here, I'm showing almost a wine bottle-shaped patch. It's covered in these RPE cells. And on the end, there's something there to try and help the surgeons place this in under the retina. On the left, you can see an actual eye that's had this patch transplanted underneath the retina in the area of atrophy, or the area they can't see. So what were the findings? Well, the imaging showed that the RPE survived. Not only that, it suggested that the photoreceptors seemed to improve after the RPE transplants, and there was integration and a link between the two, which is very important. None of the eyes had any problems with vision loss, and in fact, one eye improved by a couple of lines. So this suggested that, again, it was safe, and that there was some minor improvements in vision. And remember, these are very early trials with quite severe stage patients. So you'd expect that maybe results may be better with patients who have better ventral vision. So most recently, they've given a further update, and they've now got 16 patients with a median age of 78 years. The surgery itself takes almost 3 hours, but is very variable between 2 hours and almost 466 minutes, so very variable in terms of time. But essentially, these are very interesting studies, and the next stage studies are really
with another group, which is also the National Eye Institute, which is using a similar kind of technique. But what they're having is, rather than a permanent patch with the plastic, which stays in the eye, so you have a biodegradable patch. So essentially, they'll just leave the cells in the right place. On top of that, they're not using embryonic stem cells, as these studies have been, but are using the patient's own stem cells, or induced pluripotent stem cells. And as a result, they bypass a lot of the hurdles and problems that you have with embryonic stem cells. So really, this area is probably the most exciting, and I think in the next year or so, we should be finding the first results from that first phase 1/2 study. And the phase 1 study at National Eye Institute, or phase 1/2 study at National Eye Institute, should be starting quite soon. And really, we should be looking for the next couple of years about moving forward to further studies if visual improvements are maintained. So, I think the next stage to think about is, and these are the next stage studies that are coming in preclinical, are the fact that I've shown you RPE transplants, and they're really effective when the RPE is gone. But the photoreceptors, probably after a couple of months, maybe a couple of years, they start dying very soon after. And so really essentially what's required is a sandwich transplant. And this has photoreceptors, RPE, and maybe a small blood vessel layer underneath. And so this is work that's, I think, ongoing and being tried in preclinical studies. So very few studies of these have been published. So I'll hopefully be able to give you an update of these kind of studies in the next year or two. So if you could make those sandwich transplants, that could really help not only with macular degeneration, but other diseases where the inner retina is intact. So such as retinitis pigmentosa or retinal detachment. So let's move on to case 2. So this is a 50-year-old patient who has poor peripheral vision. Has nighttime vision problems and also peripheral visual constriction. Central vision, however, is relatively good at 20/25. So I'm just showing some images here of a pale optic nerve, a loss of blood vessels, and also pigment changes everywhere, suggesting degeneration. And this is a case of retinitis pigmentosa, and the name pigmentosa really comes from this pigment. So RP, sometimes it's called. So the vision with RP is very different from macular degeneration. So macular degeneration affects the central area. Retinitis pigmentosa gives you peripheral tunnel vision. And so let's talk about stem cells in this condition. So in this, there have been various different trials of supportive treatments. And this may potentially stop any degeneration. So the trials are occurring in retinitis pigmentosa, but they may help in other degenerations as well. So at the moment, there's 2 main trials which are in phase 2. And the first one is J-Site, which is just up the road at UC Irvine, and Reneuron. And I've just been in contact with the J-Site team because they've had some good results, which I'll talk about just now, and they're going to be moving to phase 3. And so hopefully we'll be able to move some of the patients from our clinics and suitable patients to this trial, as Irvine's very close by. So how does it work? Well, I said it's phase 2B trial. There are retinal progenitors. That means these are baby retinal cells from stem cells. And so as a result, they're growing and they're releasing chemicals. And these chemicals or growth factors can potentially help other cells which are having trouble from disease. So it's thought that this may delay or actually improve dying cells. These cells are transplanted into the jelly of the eye and where they release their growth factors. So what are the results? So I'm just trying to summarize the complex of findings from the study. There were 74 patients looked at, and they had increasing doses of cells. So there's a placebo, a low dose, and a high dose. What they found in the initial results of the 74 was that there was a mild increase in central vision, but this was not significant between the baseline and the highest dose. That means there wasn't a clear result. But what they found later on was that certain patients were at a slightly better level, which means they still have photoreceptors in the central area, which and they're the things which are important, or the cells which are important for central vision. So when they took the patients with good central fixation and photoreceptors, they found 37 patients. And in these patients, they found a significant increase in vision with dose. And these are the studies that really supported the phase 3 trial, which really should be starting later on this year. So again, this is quite exciting. If this works, this could potentially help a number of diseases. But we'll have to wait sort of really before we find the results of this study. And the other trial, which uses similar progenitors, but they're transplanted under the retina, is the Retinal study, which is based in France and also in Harvard, or Mass in the air. So that was a supportive or paracrine cell support with stem cells. So let's move on to the final set of things, just a bit wary of time. So final set is, so this is case 3. And again, this is a patient that I've seen in one of my colleagues' clinics, but 65-year-old patient, otherwise fit and well, feels that things are not as clear as they previously were, generally in terms of contrast, and also occasionally bumps into things. So again, has lost some peripheral vision. So not unlike the case before, but slightly different. Again, there's a slight family history of conditions, and there's a raised intraocular pressure. So fluid in the eye, this is the front of the eye. So I'm just showing a picture of the lens. This is the ciliary body, and this is drainage. And fluid is generated in the back or middle portion of the eye and goes to the front of the eye. And due to various mechanisms, what happens is the pressure builds up inside the eye. Eventually that causes damage to the nerve. Here's a very white nerve showing damage. It should normally be pink. High pressure inside the eye can cause damage, causing glaucoma. And glaucoma again causes variable visual field differences, which can affect central vision and also potentially peripheral vision. So this set of treatments can potentially sort of treat glaucoma and other optic nerve disease, so optic neuropathies, optic atrophy, and optic neuritis, which is commonly seen in diseases such as multiple sclerosis. So this is not my area of specific research, but I think it's very important to update on the developments of this. And one of my colleagues, Jean Doe, is doing research on this, sponsored by the NIH. And so let's talk about the optic nerve. So essentially, this is the eye at the front, and I'm just drawing or showing the pathways down the optic nerve. There's a long cell that goes all the way from retina into the middle of the brain. And so what will happen is if there's glaucoma, this nerve or long nerve is damaged. So this has to be replaced. So it's a very long pathway. And this is essentially why it's thought that glaucoma occurs, because this nerve is very long and also very susceptible to damage. So this is a preclinical study, which is published in 2016 by someone who was previously here, Jeff Goldberg, who's now moved to Stanford, who's the chair there. But some of this work is now being carried on by, as I said, one of my colleagues here. So what they did was they looked at rat retinas, and they transplanted labeled retinal ganglion cells, so RGCs are called them, retinal ganglion cells. They're the cells which are in the inner part of the retina that go all the way along to the brain. When they put those into the jelly of the eye, they found, you'll see in green here, the optic nerve is in the center of this picture, and the green dots are all where the transplanted cells are. And what I'm showing is long lines from the transplanted cells to the nerve, which shows that they generated nerve fibers. Looking at the cells, they look like normal cells from unaffected individuals. They have a spider-like appearance. And when you do a cross-section of the retina, you can again see the green with the long nerve fibers. So essentially what these studies showed were that the cells which are transplanted have just become like host cells, and they grow out nerve fibers which go down the nerve. So this is very exciting, because this could potentially be a regenerative mechanism for people with optic nerve disease. Now, with further information, I won't go into the details, but basically there's a number of factors which can help these cells grow, these long nerves or nerve fibers. So there's stem cell-like factors. So again, some of the things that I've shown you before is very similar to those. And the normal retina has suppressive factors which stops the cells growing, because when you damage the nerve, you don't get regeneration. And that's very important because you don't have constant regeneration, otherwise you'll not be able to see properly. But in these instances, you'd like regeneration. And so there's certain markers, chemical markers, which suppress growth. Finally, there's also other factors which can help inhibit axons forming, or the nerve fibers forming. So manipulating these can potentially sort of help the nerve grow. And what I'm going to show you here is quite remarkable results. So instead of obviously having a glaucoma model, they did a nerve crush model. So this is similar to the spinal crush injury models. So I put a star here on an optic nerve, and this is where the nerve is crushed. And you can see that there's no growth of green nerve fibers beyond that area. They've all died. What they found was when you had inflammation in the eye, say by injuring the lens or doing other things, then the inflammation caused the nerves actually to grow beyond the point of crush. And then if they modified some of those factors that I mentioned, again, the nerves grew beyond that area. In fact, when you did a combination of the right things, there's almost a full regrowth of the nerve. So again, this is very exciting because essentially it's showing that the nerves can regenerate beyond an area of damage. And this is another scan, again with a label scan, which shows the area of nerve crush early in the nerve, and then the cells going all the way back into the brain. But it's great that the cells grow. As we talked about in the retinal studies, does it have any function? So how they tested this was they took the mice who had or the rats who had the nerve crush, they transplanted the cells, and then they put them into a little maze where they were seeing black and white lines, which allowed them to see. And what mice and rats tend to do is they follow the maze, or they follow the black and white lines when they revolve around. And normal eyes obviously have a very good response. But what they found out was with the regenerating eyes, there's a gradually increasing response over 3 months, which is about the time span of the growth of these cells. So this is very promising. And these are only with certain factors. So you can imagine that if they had other factors, this could improve further. And in fact, very recently, another paper by David Sinclair and Harvard Mass Eye and Ear did a very similar thing where they did a nerve crush and then they put stem cell factors into the eye. And again, very similarly, they had a very marked improvement in terms of functional vision. So this reversed the injury, but not only that, the stem cell factors also made the eyes younger. And this is very promising research. But what would it take to translate this to clinic? Well, this is preclinical work. Mouse and rodent models, so obviously the eyes are slightly different from humans. So the next stage really is to do this in non-human primates. So that's the next stage. And then potentially in humans as well. So, and we're moving far faster now to human clinical trials. So this is very encouraging. And they seem to have figured out now the markers which allow the nerve to go back into the eye. And so when the retina is intact, such as optic nerve disease, the photoreceptors are still functional. So it's just the optic nerve that needs to be replaced or regenerated. And these things can potentially help. So just to conclude, I've done a brief run through all these things. I'll maybe speak about a bit more specific things, especially my area of interest, the retina, later on when answering questions. But hopefully I've made you understand that the visual system is complex, and which is why it's taken so long to try and heal the retina and the optic nerve. There's lots still to understand, but there's been a lot of progress, especially with the patch side of things and recently with the optic nerve. It's hoped that these changes will hopefully coming to clinic, and especially the RP trials, they're in phase 3, and the supportive treatments, they're in phase 3 now. We're hoping that if they're success, then the next stage will be clinics in the next couple of years. with preclinical studies, obviously they'll take a bit longer, but I think the results are very promising. So thank you very much for listening. I'm happy to cover any questions. I'd like to thank a couple of people, especially some of the professors who have helped me in some work from the UK originally, which I've brought now over here to UCSD and Charlie Eye Institute, and also the sponsors, sponsored by the National Eye Institute's National Institute of Health and Foundation Fighting Blindness, who sponsored me as a Career Development Award, and also the Wellcome Trust. So we're very fortunate to have funding to be able to do these studies, which are quite time-consuming and expensive. So thank you again for your time.
Wonderful. Wonderful. Thank you.
So I'm just going to unshare now.
Perfect.
And now we see you again. At this time, we have opened the microphones, and if you have a question, please raise your hand in the app. And I'm going to ask Bradley Day if he would handle the questions in the order that the hands are raised.
Thank you very much, Wanda. As mentioned before, for the questions, please do try to keep them brief and general as possible, so that way all can learn from the questions that you pose. And as mentioned, if you are able to use the raise hand feature, then we will call on those who have their hands raised. We'll go until about 5 minutes to the hour and see where we are and go from that point. We'll start with Lori Patterson, who had her hand raised, and just so you know, you're muted at the moment. So please unmute yourself, Lori, and share your question.
Hi, thank you so much for making this presentation. You explained about the different time, how long each phase is, and can you please tell me how long again is the phase 3 trial? And when do you actually see something viable coming on the market that will for sure work?
Yeah, so thanks, Laurie. I think these are questions that my patients constantly ask. So I can understand we're frustrated because we can't do anything, and obviously you're very frustrated because of the vision. So for phase 3 trials, it depends on the disease, obviously. So with the disease such as macular degeneration, the changes are relatively slow. What pharmaceutical companies who are invariably involved in these studies, they need to do is get markers within at least 2 to 3 years. So really, in the current diseases that I'm treating for other studies, so I've got I brought about 10 different trials now to UCSD, and I'm also co-investigator on a couple. And one of them is a GA, or geographic atrophy, dry AMD trial. And really, they want to look at the outside part of the retina and see how fast that's growing. So that's going to be about 2 to 3 years, another year or so for analysis, and that's where we find out the results.
Wow. Very informative. Thank you for that question. We'll now turn to Emily, who has her hand raised.
Yeah, fine. Can you hear me?
Yes, we can hear you.
My question is about optic nerve stroke, and I heard you say that it's studies. So I'm just curious.
Sorry, Emily, I'm just having trouble hearing you at the moment. Sorry.
Your volume. Can you increase your volume, please?
I don't know if I can do that. Does this help if
That's
right
better.
here?
That's better. Yeah, thank you.
Perfect.
Okay. Yeah, I'm curious about optic nerve stroke, and if you have just approximate a time period where the research, because I know you said it was new, and so I'm just curious how much longer.
Yeah, no, sure. So listen, I think one of the things I should really sort of talk about is really the phases of the trials, which I think you brought on to. So preclinical can be, depending on the type, 5 to 7 years. Some of those studies may not get to clinical. That's where they put it to the side. Now, these studies have got quite advanced now. They're probably maybe 2 or 3 years away, if they're successful, to the next, maybe a clinical trial. There are potentially one or two other steps, because, as I said, rodent models aren't the same as humans. So you really want to try and do this in a more human-like condition or model. So the question is whether they do that or they go straight to human. And as I said, there's the FDA and others are more likely to accept human studies nowadays. So that's one thing. Then you're looking at the progression in each stage that you're trying to measure. So you can see that it takes weeks, months to years for the cells to grow. So first of all, in phase 1, you're looking at safety. So phase 1 trials, yes, they're small in number and they're relatively quick, but they have to have a safety. So that's probably about 2 to 3 years. It varies again on the disease and the model, and then they analyze that. They go into phase 2, or sometimes they have phase 1 and phase 2 together to try and speed things up. And again, if it's phase 1 and 2 together, it's probably about 4 to 5 years. And phase 3 is about 3 years with analysis. So you can see that this adds up, and the reason is because they have to show efficacy to the FDA at the end, obviously, and these diseases, some of them are very slow, and recovery is very slow. So hopefully that gives you an idea.
Okay, thank you.
Thank you for that question, Emily.
Bradley, I think the next person, her name is Candy, if I'm not mistaken.
Okay, thank you very kindly. Yes, the next person then by email, assuming the name is Candy, you may ask.
It is. Thank you very much, Doctor. That was great information. I have a couple of small questions that shouldn't take too long. Are there other countries that are further ahead than the United States in doing stem cell research? And how do you feel about stem cells, that's what you're in, but also versus gene therapy for retinitis pigmentosa?
Yeah, so those are great questions. So one of the great things about global development, and I'm an example of this, is I started my research in the UK, but I came over here to look at a specific model of research, and we were getting cross-fertilization of researchers around the world. So essentially, at certain points, certain countries have got further. Maybe, so for instance, the UK at some points was maybe further, or parts of Europe in terms of big clinical trials. But then America's caught up with the latest trials. But essentially, you're one of the best places in the world to get the latest stem cell treatments, or because of the funding that you've got, because of the number of people who are affected. And really, essentially, it comes down to sort of the pharmacology companies or pharmaceutical companies, and America's got the biggest pharmaceutical companies that will drive research. So there are other places around the world who are doing research, but essentially, you're in the best place. You're in the front line. Now, the second thing is the retinitis pigmentosa, and one of the main things I brought to UCSD is gene therapy trials. And so gene therapy is effective when there are cells which are there, but if cells aren't there, then you can't do gene therapy on anything. And so it's really a different stage of disease that we're treating, really. And so what I've been talking about today is really late stage or cell replacement, regenerative approaches. And so gene therapy, I think, is fantastic. We've had some great results with children that we've had, but I think it's a different stage of disease, yeah.
Thank you.
Wonderful, wonderful questions. We'll continue onwards with Kelly, who has a hand raised. Kelly, you may unmute yourself and yes, answer your question.
Yes, thank you. Hi. So I am just wondering, for someone who has a huge hole in the macular area, which is not able to be repaired surgically, would that be a potential situation can be helped by stem cells to regrow that area in the macular area? Thank you.
Yeah, so macular hole is a very relatively common condition. So in the retinal clinics, you'll probably see 2 or 3 people a week at least with this in UCSD. And that happens when the jelly pulls away from the eye and leaves a hole. So essentially what's happening is there's a hole of the whole retina there. So not only the RPE, but the other part of the retina. Now recently, and again, you may be able to ask some people, but they've tried to plug those holes with recent techniques such as amniotic membrane to try and get the retina together again. And that seems to have improved vision anecdotally in a couple of cases. So maybe something you may ask again about. And the amniotic membrane also releases some factors as well. So that may be helpful. And if it's the RPE and photoreceptors that are still lost there, then maybe at a later stage there could be a chance for
for that kind of transplant, but that's much further down the line. So the first stage is really to get the retina together. So maybe it's worth just asking about your vitreoretinal surgeon to see if there's a sort of more latest kind of version to try and get the whole back together again.
Thank you. We'll take time for about 5 more questions or so, since we do see that those hands are up, and we'll ask to keep them brief. We'll go a little bit over so that way we can answer everybody possible. Moving next to Yolanda, you may unmute yourself and ask your question.
I just had a quick question. My mom actually had a tumor removed from the brain, and she became blind after the surgery. Would this be like a possibility, like in the future for her, like to be treated with stem cells? Or yeah, I don't know.
Yeah. So again, there's a range of diseases I talked about today, and there's probably a range of diseases that I haven't really talked about. The diseases that I talked about in terms of the optic nerve, I said it's a very long pathway fiber, and that treatment actually is very supportive of other nerve abnormalities. So the visual pathway, as you know, as I was explained, is very long from the front, and for some reason the processing is right at the back of the head. So it goes all the way to the back of the head. So I suppose the question is, where the damage has occurred. So if the damage has occurred more at the front of the eye, in some kinds of tumours that occurs, such as pituitary tumours, then this could potentially help. In ones which are further back, the research there will probably need a bit more in terms of brain research, but there's potential in terms of pathways. So I think really the question is where that is. But remember, the first thing they're going to do is probably treat the simpler cases first, and then when they become treatments, then obviously they'll try that. They'll be available to other people.
Thank you.
Thanks, Yolanda.
These are fantastic questions. Thank you all. We'll turn to Mr. Gregory Reed. Thank you for your patience. You may unmute.
Thank you, doctor. I really appreciate your presentation today. I'm afflicted with Usher's 2 and glaucoma.
Mm-hmm.
I'm very interested in what you shared in regard to regenerating nerve stem cells. Is there anything in the testing or studies in regard to sensory neural deafness? Are they working with regenerating nerves for your hearing?
Yeah. So Usher syndrome is one of the areas I sort of subspecialize in. Fortunately, we have a retinal degeneration clinic there, and I work very closely with someone called Elena Carey, who's a specialist in hearing. She's based mainly at Radies, but also at UCSD. Actually, one of the labs I was at previously in London, run by Robin Alley, it's a very bright postdoctoral scientist there who's now a lead researcher in Sydney. So her and a couple other people around the world actually are developing. We've studied not only the retina, but she made retina, but she also made ear cells. So they're definitely looking at this. But in your case, I can hear that obviously there's some hearing deficit, but what you want to do is optimize the hearing that you've got. Especially the Usher type 1 patients, this is the case where you may have to do something slightly different. But there is work in terms of regeneration. So I'd look at those avenues first in terms of the eye and ear clinics before we jump to stem cells.
Fantastic. Doctor, to be respectful of your time, would you have time to answer about 3 more questions?
Yeah, I've probably got another 5 minutes, if that's okay with everyone. Yeah.
Wonderful, thank you. Dennis, you may ask your question.
Okay, so I'll talk fast. So I was a pioneer, one of the patients from Scapins, Okamura. Scapins did the hot treatment for welding retinas. Okamura went to cryo, and McNeil went to the laser. So I've had all 3 of those done on me. And I've lost my vision. It's degenerative. And I have a pale optic nerve. So the retina has been stable since 1969. The optic nerve is where I'm concerned. Do you think the things you're doing with retinal will translate to the optic nerve?
Yeah, so I purposely did my talk in different stages. So I talked about the retina and the outer retina. I did another talk about these supportive cells. The final part of the talk was about the optic nerve. So the optic nerve studies really are, in probably your case, what you're describing. If the retina is stable on imaging, it looks like there's normal retina with photoreceptors, and the damage is really in the retinal, that means the optic nerve, then those last set of studies were the ones that you want to really be focusing on. And I said, so there's different groups, but one at Stanford and also one which we've started here with Jean Doe, one of the glaucoma specialists, who's focusing on trying to regenerate the optic nerve.
Thank you.
Thank you. Raymond, would you like to answer or ask a question?
Who'd you call?
Raymond. Raymond.
Yeah, that's me. Hi, Doc. How you doing?
Yeah, hi, Raymond.
I've had the retina for about 14 years, and I got the wet type. How soon do you? I'm 76, and I've been stable for the last 5 years. I haven't had a shot or anything. How long do you think it'd be before they could, maybe experiment on me?
So just stepping back, you've got wet macular degeneration. So you're one of the relative lucky ones. Okay, so because there's currently a treatment which is very good for you, and so the main thing for you is if you have a sudden change in vision, to pop to your eye doctor and then have that treatment, which is a little injection, as you know, probably. So that has been shown relatively over time to keep most of the eye stable. After about 7-8 years, there is some ongoing macular degeneration, and then in that case, that would fall under the dry type degeneration that we talked about. But really, I think your main trick is really to just keep watch on your eyes and then go to the doctor as soon as you can to get these treatments if there is a change. Because what we find is there's only about 10% of patients who probably don't ever need an injection again. Usually with wet macular degeneration, you will still need an injection from time to time.
Okay. Thank you.
And then the last question, just to, if possible, is Barry. Has their hand raised?
Thank you very much for doing this. It's excellent, absolutely excellent.
You're welcome, Barry. Yeah.
I would like to ask you 2 questions. One, is the RPE patch currently available? Is it still in trials? What is with that? And secondly, do you work with Dr. Sada at Doheny?
So taking the first question first. So the RP patch, as I tried to mention, I think is in phase 1/2 study. So that means it's basically a safety combined with a efficacy study. And so that means that study is still ongoing, and that analysis should hopefully be later on this year or early next year. COVID has obviously, as you know, put a real hamper on or hampered these studies. So that's in trial rather than treatment phase at the moment. But as I said, the early results look promising, and they've also shown great surgical results. So that's the patch side of things. Dr. Sada, yeah. So Dr. Sada is, I don't work with him directly, but I mean, he is a great imaging expert. So he deals with the retina at the back of the eye, and he's very well known, Doheny and UCLA. So we know of him. My colleagues have worked with him, and I've been to visit him, but haven't worked with him directly.
Thank you, Doctor. Thank you for all of your questions. We'll turn the time to Vicky for some final comments.
Okay, well, it's that time again. Unfortunately, we could go on, and we appreciate everything. We really want to extend our gratitude to Dr. Shyamanga Borooah for taking time out of his very busy schedule. You have no idea how busy that is. I don't think any of us can understand that. So we're very, very grateful for the time that you spent to educate us. We're also very appreciative of everyone who was joined today to gather this information, to educate yourself. And please know, as Wanda mentioned, we will be sending you information regarding Dr. Borooah's practice and how to contact him, not for follow-up questions, but if you'd like to see him as a patient. So instead of goodbye, I hope that you will follow up with some of our other programs that we have at Braille Institute. They're all going to be virtual, but we pretty much offer everything that we were offering before COVID. So please stay in contact with us, and we look forward to seeing you at another presentation. And thank you again.
Oh, Doctor, did you have a? Looks
yeah,
like
I'd just like to finish. Sorry, thanks, Wanda. So thank you very much, first of all, for this. I know COVID has been a real struggle, especially for the low vision community, and I'd especially like to thank the Braille Institute with Jay and Vicky. And without them, I can't deal with some of the resources and community work, which they really follow up on with my patients. So thank you again for listening, and I hope to see some of you soon. Yeah.